Thank you very much for this introduction, and thank you for inviting me to speak at your conference. This is my first time. I wish I was able to be there in person, but unfortunately, because of COVID and other family matters, I'm unable to be there. But I will try to share as much information on the subject of potato early dying with you in this 40 minutes or so. So potato early dying research update and disease management is the topic of today. So potato early dying, which I will refer to it from time to time as PED, is a complex disease, and it's a vascular wilt disease which results in premature vine senescence, and it can limit potato tuber yield by as much as 50%. Primarily, it's caused by the fungus Verticillium Dahlia, but the co-infection of potatoes by both Verticillium Dahlia and the root lesion nematode, which the scientific name of it is Pratylenchus species, can greatly increase the severity of the disease. Now, if you look at the pictures here that I have, the one on top right, it's like a greenish tissues with small black dots on it. These are the microsclerotia of the Verticillium fungus. That's how the fungus overwinters during the season where there is no crops. And on the top left, you see a root that has worms-like structures there. These are the nematodes attaching themselves to the root of the potato plant. And on the bottom is a field that is heavily infected with potato early dying that shows the symptoms. So how does Verticillium survive? Verticillium is one of the two culprits. It's a Verticillium and the nematodes cause the potato early dying complex. Verticillium can survive both in the soil and in plant tissues. In the soil, it survives as microsclerotia, and these are the ones that I mentioned a minute ago. They are resting spores, overwintering structures, as you see on the picture on the left. Or in the plant tissue, it can survive as mycelium, which is another structure of the fungus, or microsclerotia in both in potatoes and the tubers themselves, the leaves and the stems, as you can see in the picture under there on the right hand. In the middle, you see a potato leaf that show typical symptoms of Verticillium wilt. So how does Verticillium affect the plant? It invades the xylem tissues, and it disrupts water transport and supply in the plants. And it causes foliar discoloration, necrosis, which is death of the tissues, and also vascular wilt. And it persists in the soil as microsclerotia for a long time, for more than 10 years, in the absence of the susceptible host. And once you plant a susceptible host, whether that is potatoes or others, which I'll show you later, then it will reactivate itself and infect these crops. Once it's introduced into the field, the fungus spread rapidly by runoff water, cultivation and machinery such as harvesters, which transport the soil which has the fungus in it or the nematode from one part of the field to another. Literature had put an economic threshold for Verticillium when we test the soil or the plant samples, and it varied. There is some that say it's between 5 to 30 microsclerotia per gram of soil, and some go with a narrower level of 8 to 12 microsclerotia per gram of soil. Verticillium has a wide host range. Almost all dicotyledons, which are broadleaf plants, are susceptible. And that includes potatoes, of course, alfalfa, clover, sunflower, cabbage, celery, cucumber, pepper, radish, rhubarb, tomatoes, eggplant, mint, soybeans, and probably others. However, monocots, which are cereals, and also forages, so cereals such as barley and oats and wheat, and forage grasses are not hosts for verticillium, but they are hosts, later on you'll see, for the nematode. Asparagus and lettuce are moderately resistant to verticillium, but none of the potato cultivars that are present and used in Canada is fully resistant or tolerant to potato early dying. So you're in the field, you want to look and see, if you can find early dying. When it comes to verticillium wilt, what to look for in the field. Basically, 4 things you should be looking for. One is yellowing of the plants, wilting of the plants, flagging, and discoloration. Now I'll go through each one separately to discuss them more broadly. Yellowing, usually the first symptoms appear immediately after flowering. So sometimes it depends on when you plant, August or so. They start in the lower leaves, and then the area between the leaf veins begin to yellow. So between the veins, you get yellow color like you see in the pictures here up top. Later, the symptoms move upwards to the younger leaves. So it starts from the bottom, older leaves, to the younger leaves up above. So initially, one side of the leaf turns yellow. So I put a rectangle on that picture with the blue background there to show that almost half, the right half of the leaf is infected. It's yellow and it's wilting, but the other half is intact. You see that in the second picture on the left, same story. One part of the leaf is wilting, yellowing, the other half is not. But later on, the entire leaf turns yellow. So this is in the beginning of when the symptoms start appearing, they start that way. The leaf yellowing eventually becomes browning and necrosis, which is death of the tissues. So the whole plant might die and turn brown in color. A question was asked about where you see verticillium. Does verticillium stay in the roots? Is it in the stem only? Is it in the leaves? We did actually do a small experiment where we took those leaves that, like you see on the left hand up top, where they show typical symptoms of verticillium. And so we took the petiole out and we cultured the leaves, as you see on the bottom left, on a specific medium. And sure enough, the leaves had verticillium in them. So it does transfer in the plant system, and you can find it in the leaves too. Wilting is, of course, another symptom of early dying. In early stages of the disease, not all stems from the same plant show symptoms. Sometimes one stem shows symptoms and the rest of the plant looks healthy. And usually you see that during the day. So infected plants wilt during the day, but they recover at night because it's cooler and there is moisture there compared to the daytime. The other important issue when it comes to wilting, what it causes early senescence, which means the plants start dying early, maybe 3 to 4 weeks before the tubers reach maturity. And I've seen that this year actually with some of our growers in the variety Shepherdie, where the plants died 3 weeks before, the end of the season. What that means is that the tubers do not size, and you get a serious yield reduction because of that. Flagging is another unique symptom of early dying and verticillium wilt, where you look at the field, see the one on the left hand, Most of the plants look okay, except one is erect and it's wilting. And that's so erect upright, and that's a unique symptom of early dying verticillium wilt. Discoloration is another symptom. The discoloration usually is in the vascular system, as you see on the picture on the right hand. This is a stem that's cut, and you see brown discoloration in the vascular system. Now, if you pay attention in the field later in the season, you may see what I saw in the picture here, some stems that have black dots on them. You may think it's a black dot. Black dot is another disease of potatoes, but surely it can be verticillium wilt. So if you cut one of the stems that are still greenish, you may see those brown discoloration, which are typical symptoms of verticillium wilt. Now, in my case, just to verify whether I'm looking at verticillium wilt or something else, we did take some of the stems, as you see in this picture. And you see on the left hand where I have the arrows there, this is a stem that is infected and it has microsclerotia already on it. So we took some of those tissues, we culture them on specific medium, and sure enough, the fungus start growing. And you see that typical on the bottom side is the fungus. That's verticillium. Typical structure of verticillium, it looks like 3 fingers, and there's balls on them or circles. That is verticillium, different species of verticillium. Tuber discoloration is another symptom. So in the tuber vascular system or ring, you can see that the tissues turn brown in color. That brown discoloration usually starts at the stem end, but it doesn't go too far. It doesn't extend usually more than halfway through the tuber. So the first culprit of the potato early dying complex is the fungus, which is verticillium. The second culprit of the disease is the root lesion nematode. And these can be present in the soil, in the roots, and the tubers. And nematodes, for those that don't know what nematodes are, they are microscopic roundworms. In fact, when you look at them under the microscope, they look like snakes, but you can't see them in the naked eye. You have to have a microscope to be able to see them. So they feed on the plant roots, and they also live in the soil on plant tissues or debris. And that's what you see on the top left picture. And so when they do start feeding, they eventually go inside the tissues, as you see in the picture in the middle, and they will block the tissues and cause problems. So they damage the root system, and they cause stunting, yellowing, wilting, poor stand, and yield and quality reduction. Now, the symptom severity is nematode density dependent. That might be a long sentence, but what I mean by this is that the larger or the higher the number of nematodes per kilogram of soil, the more severe the symptoms will be. So when you do test for nematodes and you find large number, that means the severity might be higher. What do also you look for if you want to see the symptoms of nematodes is the lesions. That's the name of the nematode, root lesion. So it causes lesions on the roots. They are reddish to brownish to start with, but eventually they turn dark in color, as you see in the picture with the blue background on the right hand. Now, with most plant parasitic nematodes, we know that, those that live in the soil, and lesion nematode is one of them, they do not move very far. So they don't migrate more than 1 to 2 meters from the root zone that they infect. So it would be within 1 to 2 meters from the plant root system that they infect. Nematodes also have a wide host range. Grasses and cereals are one of those. Field and vegetable crops and weeds are also hosts. And if you remember, the cereals and grasses were not really hosts for Verticillium per se, but they are for nematodes. The preferred hosts for the nematodes are potatoes, corn, carrot, sugar beet, vetch, soybean, raspberry, and strawberry. However, canola, forage pearl millet, Sudan grass, broccoli, and onion are non-hosts for the nematode. They are naturally occurring in grasslands and pasture. You can find them there too. And the economic threshold for root lesion nematode is set in literature to be between 1,000 to 2,000 nematodes per kilogram of soil. And the 1,000 is for the most susceptible variety of potatoes that is superior. And the 2,000 is for the rest of the potato varieties. So that's the threshold, the economic threshold. Okay. Now, research update. I thought to show you some of the research work and the results we've done in New Brunswick. I've been working on verticillium wilt and potato early dying since I think 2015. We were the first to start working on this because we've seen some symptoms in the fields. So we've conducted field surveys in New Brunswick since 2016. And that season, I surveyed 163 fields after harvest. And then 2017 and 18 and 19, we had also followed up and did soil sampling too to see what the status is, because we really didn't know what's going on and what the levels are in potato and other fields on farms. I thought to put this slide because it's quite important, because you can take soil samples, and if you don't take them correctly, then you're not going to get the right results. So soil sample collection is really important. What we usually do is we soil sample following zigzag pattern, and we used a soil probe that is 2.5 centimeter in diameter, as you see in the picture on the bottom there. Sometimes I used bigger ones. It depends on how much soil I need. But we drive the auger about 25 to 30 centimeters deep in the ground, or up until we can't go any further. And we take soil sample. We usually take a number of soil samples from each field, depending on the field size. But generally speaking, if we have the staff and time, we take one subsample per 10 feet. And these subsamples are emptied in an empty clean plastic bucket or pail, as you see on the bottom picture. We take usually 2 kilograms from each field. We mix them really thoroughly. Half of that is used for testing for nematodes, and the other half is used for testing for verticillium. Sorry, I'm trying to use the errors for the PowerPoint, but they're not working, so I'm trying to use another methodology here. For verticillium extraction and identification, the soil has to be dry. So we air dry the soil, as you see in the picture on the bottom far left. So we put the soil on trays, we air dry it, and then we sieve it, as you see in the picture next to it, in a 2 millimeter diameter sieve, and then we freeze it. That way we can test it anytime if we don't have time to test it immediately. Testing is done by different methodologies. One of them is molecular testing, and we use RT-qPCR, where the DNA is extracted and the test is performed, and the cell number of Verticillium species per gram of soil is calculated or estimated. We do this for both Verticillium Dahlia, which is the more dominant one species of Verticillium in our area, and also for Verticillium alboatrum. The second methodology to test for the Verticillium or the for the Verticillium itself is by culturing the soil on specific media called NP10. And I prefer that method because that way then you get number of sclerotia or colony forming unit, we call them, per gram soil. And that's what you see on the bottom third, fourth, and fifth pictures, where the soil is cultured on the media, and whatever grows, that means it's active. When you do molecular analysis, whatever you get doesn't mean active because you're extracting the DNA, and that's what you're testing. For nematodes, for root lesion nematode, we don't dry the soil. The soil is used as is, wet, not sieved or frozen. We collect it. We send it. Usually, we send it to the University of Guelph for testing, and they test the nematode, the sample, and identify the type of nematodes they find, and also the number of nematodes per kilogram of soil. The nematodes like water. That's why they have to stay moist, and if you dry them, they're gonna die. So when they analyze them, they won't be able to detect them, because what they do, as you see in the picture on the left hand, they put the soil in a tissue and water underneath it. And nematodes are hydrophilic; they love water, so they swim towards the water, and the water is collected and tested for nematode. These are some of the results from previous work. So in 2016, we had a large number of fields that were surveyed and tested for nematodes and Verticillium. And the fields were planted with various crops. And sure enough, if you look here, you would see large number of root lesion nematodes in corn, potato, clover, oats, and so on. And these are basically the hosts for the nematodes. In case of Verticillium Dahlia, the largest number of cells of Verticillium were found in the potato crop fields, ryegrass, and clover. The rest really were very low or non-existence because they're not hosts for the nematode. Grid sampling is important here. And I mentioned grid sampling because that's what growers usually use when they do test for nutrient content for the soil. We did some work on grid sampling to see. What I wanted to show actually in this slide is if you were to pick some samples from the field and you don't do it correctly, and you are going to end up with the wrong results. For example, if we pick WC Spring 218 field, we took here samples from 8 locations. Now, if you only took samples from location WC8, you only will find a little bit of Verticillium. If you took it from WC2 here, it's clean, there's nothing. You may think my field is fine, nothing in it, and that's incorrect. But if you look at WC7, WC8, you find high level of both Verticillium, which is in the blue color, and nematode, which is in the red color. So the best thing is to take a random sample from across the field and mix them together, and that gives you the best results that you need. Here also, I wanted to show on this table what happens over time. We did follow some of the fields over time to see do the numbers of Verticillium cells or root lesion nematodes change in the fall and the spring when should you take the soil samples? So for example, here in the fall 2017, the number of verticillium cells per gram of soil were high, but when we took samples from the same fields in the spring, they were low. Same thing for root lesion nematode. So if you take the samples in the spring, you may think you don't have a problem, but actually it's because either the nematodes are going too deep in the ground because during the winter they hibernate, they go far away from the freezing and so on. And same thing for verticillium, the numbers will probably be reduced during the winter time because of killing them by freezing, but they bounce back in the fall after the crop is planted. I'm part of a big network called Canadian Potato Early Dying Network. We have more than 20 scientists working on potato early dying from all aspects. So one of the aspects is to see the efficacy of various products that are available in Canada, see how effective they are against early dying. So this experiment was supposed to be repeated in various provinces. I think it's in 5 or 6 provinces. So we have a control where there's no fungicide added. Elatus, which is a new fungicide added in farrow for the control of verticillium wilt. Vellum prime is a nematicide for the control of pathogenic nematodes. Serenade soil is a biological control material made of a bacterium called Bacillus subtilis. The company and the industry felt that we should try this product as they feel it's effective. Senator PSPT is a seed piece treatment. I did try it just to see how it works. It's registered for the control of verticillium wilt, but it might be being phased out by the industry anyway. We also added 2 treatments where we had elatus and vellum. We wanted to see if we put both together, would they have a synergistic effect? And also added serenade and vellum and elatus to see if there's any synergistic effect there. The potato variety we used was Russet Burbank. Now in New Brunswick alone, I was able also to test the product Vapam, which is a fumigant, on a field scale. So there's a grower that wanted to test this in his farm. He wasn't able to apply it in the fall, so he applied it before planting, about 3 weeks before planting, 28 gallons per acre he used. He said it cost him about $390 per acre. So he had split fields, and we had 2 different varieties. One was Russet Burbank, one was Snowden, and we tested to see how they work. So these were the results from 2020. This shows the effect of different treatments on the number of root lesion nematodes in the potato field. And it's the percent decrease in decrease in number of nematodes relative to the control is shown in this picture. Before treatment is in blue, after treatment is in yellow or orange. Vellum worked really good, so it reduced the number of root lesion nematodes by about 58%. Serenade did well too, 79.4% reduction in the nematode, and so did Senator, reduced the nematodes by 67%. In case of verticillium, elatus reduced the number of cells or colony-forming units by 53%. Vellum, also it's nematicide, but it did also reduce the cells of verticillium by 73%. And so it did the mixture of elatus and vellum, reduced it by 33.3%. Now, the major thing that we could show and tell about these treatments is their effect on marketable yield. Now, all of them had positive effect on marketable yield. Elatus alone increased marketable yield by 7.6%, vellum by 8 or 4.4, Serenade 9.6, Senator 25%. And although the mix of treatments didn't have any effect on the number of root lesion nematodes or the cells of verticillium, they did increase marketable yield by 26 and 27%, as you see in these pictures. Now, this shows the split field that the grower had for Russet Burbank and Snowdon. I'm only showing the effect on marketable yield, but I also was there taking data, and you could go to that field and you could tell which half of the field was treated and which half wasn't. The treated half was still green, and non-treated half with VPAM was dying early comparatively. So in case of Russet Burbank, the grower had an increase of about 20% in marketable yield, 5.11 tons per hectare. In case of Snowdon, it was a little bit less, but about 2.6 tons per hectare increase in marketable yield. We also worked on other products like chloropicrin, which is the tear gas. Again, it's a fumigant. And we tried it on 8 fields. We took samples, soil samples before our application and after fumigation too. And sure enough, as you see here, the reduction in root lesion nematode was high, in some cases by 99%. So the blue was before fumigation and the red bar is after fumigation. And there was an excellent results there. In case of Verticillium wilt or dahlia, the number of cells of verticillium also decreased due to fumigation with chloropicrin, with up to almost 95–96% reduction in verticillium cells or colony forming units when the fumigant was used. So that's root lesion nematode. This is the fumigation with Verticillium Dahlia. The threshold for Verticillium, the literature, is vague about it. So my colleagues, Matrish Singh, Tyler McKenzie, Dahu Chen, did some work on this. So what they did is they introduced Verticillium in microsclerotia to soil, and they planted technituber of Russet Burbank. So they started with 0, 5, 25, 50, 100, and 500, and 1,000 microsclerotia per gram of soil, and monitored to see how the plants grow. And sure enough, as you see, 105 days after planting, the 5 microsclerotia reduced above ground by biomass by about 30%. And 1,000, you can see the plant is barely living there. So that was a good indication of what to look for. Sorry, conclusions, soil treatment with fumigants, chloropicrin and VPAM, and also the fungicides Elatus and the nematicide Vellum Prime reduced the level of verticillium and the nematode in potato fields. Soil treatments increased marketable yield, but the levels were variable, as you saw in the slides. And some of the graphs also showed that rotational crops are hosts. And 5 microsclerotia per gram of soil was enough to reduce the biomass, above ground biomass, by 30%. Growers should soil sample their fields properly, like I showed, how you take the soil samples and where in the field, not only one side of the field, but the whole field. It's important to identify what pathogens are present and in what quantity. And this will give them an idea on what to use of products to manage the disease properly. Finally, how to manage potato early dying. This is my last slide. There are different methodologies or practices that the growers can use. One of them is cultural control or practices. So it's important to plant clean, disease-free seed. That goes for every disease of plant disease ever. If you start with clean seed, then you eliminate the source of inoculum. Removing crop debris is important too, because as I showed you in some of the slides, the stem would have the microsclerotia in it, and that's the overwintering structure. So if these stay in the field, they will stay for 10 years until you plant a host that's favorable and they will infect it. Use long rotations with non-host crops because the microsclerotia can remain viable for a long time. Increase the soil organic matter is important. Successive years of non-host green manure decrease the disease severity. And planting Prassicaceae, such as mustard, can decrease the soil population of Verticillium Dahlia when used in a crop rotation as a biofumigant. By the way, once mustard is cut and incorporated in the soil, it needs moisture to activate, and then it produces the fumigant. And the fumigant is isothiocyanate, which is actually what's in the fumigant chemical VPAM is isothiocyanate. So you can call VPAM maybe mustard gas sometimes. High organic matter can suppress Verticillium Dahlia by either releasing toxic compounds into the soil or by improving the soil microbial activity. Host resistance is another option, but there are no resistant potato cultivars available to early dying. Gold rush, russet, ranger russet, they are tolerant to verticillium. That means they're susceptible, they may show symptoms, but they don't suffer yield loss due to Verticillium Dahlia. And finally, chemical control, as I showed, like the fungicide Elatus and the nematicide Velamprime are important. They do work. We have them available in Canada, and chlorobicrin and VPAM do work. However, they do reduce the potato early dying symptoms. What we noticed was that over time, that verticillium can build up. It doesn't get eliminated totally, but the amount is reduced. But then the next season, the level of it builds up again once you plant potatoes or favorable host crop. This work was funded through the Canadian Horticulture Council and Agriculture Canada. I thank them for this, and also I thank the agrochemical companies for providing the chemicals, and Sean Paget for the grower who let me his land to do what I wanted to do there. My colleague Dave Bell for his assistance in soil sampling and the grid sampling. Soyan Shim as my technician, she was wonderful in the field. And also my colleagues, Matrice Singh, Tyler McKenzie, and Daho Chen, who helped a lot.